I'm Professor Matt Schmidt, and this is Genetics. We're continuing our discussion of biotechnology, and in this session, we're going to focus on an analysis of nucleic acids at the molecular level. And there are a couple of sort of fundamental procedures and ideas that we want to go through in this session. The first of which is a procedure called Southern blotting. And Southern blotting allows the detection of certain sequences of interest, for whatever reason you're interested, for an extremely large amount of other DNA sequences, for example, the whole genome. Now, if you think about it, there's something in common with that statement and the screening the library procedure that we talked about if you're going in order in the last session. But we'll see that Southern blotting is a lot easier depending on your application. It allows you to pick out DNA sequences that you don't want to really do anything with. You just sort of want to get information about them. So the uses include, for example, paternity testing, forensic analysis, creating restriction maps of parts of the genome that aren't even cloned yet. We'll talk more about that in the applications section. The reason it's called a Southern blot, not because they do it down south, but the process is named for a scientist named Dr. E.M. Southern, who developed this procedure in 1975. Let's take a closer look. So how do you do a Southern blot? So let's just say you're going to do a paternity test like they do on the Maury Povich show all the time. Not to pick on Maury, but that is a pretty big part of his livelihood. So you obtain a sample of DNA, often genomic DNA, right? If you're getting it from a blood sample or something like that, and you cleave it with restriction enzymes so that many small fragments are created. Again, sort of similar to how you started making a library, right? But now is where it's going to be different. We're going to take those fragments, just the fragments. Don't worry about any cloning vectors or anything like that, and perform gel electrophoresis on your sample, which separates the targets in space. I should say the fragments in space. So we're going to talk in a second more about gel electrophoresis, but there's just hundreds of fragments when you cut up the genome like this, and when you put them in an electric field, it spreads them out so that you can have a reasonable sort of a map. It's not really a map, but you'll see in a minute. We denature that DNA that's been spread out, transfer it, you've seen this before too, to a nitrocellulose membrane, which is again basically a fancy piece of paper, and that's the blotting part of the Southern blot. And then you have a probe for your gene of interest, and you mix that with the blot, and it will bind to complementary sequences only. Then you'll perform autoradiography on the blot to obtain an image, and only the locations where the probe has bound will be visible. So sorry to keep saying this, but there are certain similarities with the procedure we use to make a genomic library. The big difference is you're not taking these samples and saving them and putting them in cells. We're just doing an analysis to see what's there. And that's, if you're doing a paternity test as one example, that's all you'd really care about, right? Now, just as an aside, gel electrophoresis is a very important procedure. Almost every technique where you're doing molecular level things, gel electrophoresis is a part of it. It relies on the fact that DNA is always negatively charged, as it's an acid and there's all those negatively charged phosphate groups around. So if you put it in an electric field, pieces of DNA always will migrate toward the positive electrode, the cathode. Now, if you just put them in water and did that, they'd migrate, but it wouldn't do you any good because you couldn't see them. So you make this gel, and it really is like gelatin. It's a matrix. You put the DNA in that gel and expose it to an electric field. The fragments will start moving, but because of the matrix nature of the gel, what I mean is if you could shrink down and look at the gel, you'd see that there are little holes in it. It's like a porous solid. So the beauty of this is that small fragments can travel relatively quickly through this maze, as it were, whereas larger fragments take a little longer time because they have to snake their way around. But it literally and almost perfectly every time, separates fragments in order of their size from small traveling the farthest to large not traveling so far. And if you use what's often referred to as a ladder, which is a marker containing DNA of known sizes, you can compare your fragments with that and get a good estimate of how big they actually are. Now, you wouldn't need to do this for a southern blot, but as long as we're discussing electrophoresis, if you wanted to just look at the gel, uh, literally look at it, ethidium bromide is an agent that we discussed a while ago. It's a mutagen, but the reason it's a mutagen is because it is an intercalating agent which gets stuck, for lack of a better term, in the DNA double helix. It gets stuck in DNA, which means it's not good to touch you, but if you stain a gel with it, it gets stuck in the DNA. And then it has this other property, is that when you shine ultraviolet light on it, it re-radiates the light back in the visible spectrum, and it looks like it's glowing bright orange. So you can literally just either hold a UV light over the gel, and you'll see the DNA band shining. It's pretty cool. All right, but back to the southern blot. So the procedure is here. So I'm putting a scenario in this picture where, let's just say it was a paternity test, and there's 3 individuals, number 1, 2, and 3. All of them are possible candidates for being the father. What I've shown is, though, in a particular DNA region of interest, and often it's not a gene, I don't know if these are showing up really brightly, but here are different cuts with the same restriction enzyme, okay? So 3 different people, 3 different patterns, and after you have the cleavage, so in these tubes are going to be floating around fragments of whatever size. Now we're going to keep them separate, separate them on a gel, do gel electrophoresis so that the smaller fragments will travel farther, but basically we're going to be separating out these fragments. Denature the DNA, because we have to do that because the probe is going to need to bind because of the complementary nature. Put it onto the membrane, and then we apply our radioactive probe to the membrane. Literally put it in a plastic bag, let it slosh around, and the probe is only going to bind to the segments that it's looking for, right? So look, guys, if you could see this, you can't see it, but this is genomic DNA, right? There's just tons and tons of fragments all over the place here. You can't see them. And the point is, the probe's going to come along, and it's going to say, oh, here's 2 places where it's binding. And that's what you're going to be able to see. And the pattern is likely going to be different for a given individual versus another. All right? So when you do the autoradiography, all you see are the fragments of interest. You don't bother getting to see all those other literally probably hundreds or thousands of fragments that are hanging around there. So you get a pattern. Now individual 3 here has a pattern that's got 3 fragments, and if you look over here you'll see the reason why. Those are the 3 fragments of 3 different lengths. Now I sort of did this on purpose, although it sort of ruins the test if it was true. These individuals in fact do have different sequences, but the way it cuts, they have 1 large piece and 1 small piece. This guy has a different large piece and small piece, but the pattern sort of looks similar there. This would ruin the test, and it usually doesn't happen because we have identified regions of DNA which are the most useful for doing this type of procedure. But the reason I put it in there is I want you to see that when you do this procedure, it's solely based on the size of the fragment. That's really all you can tell is the probe bound to a fragment of a particular size. Almost always, if you do it right, the pattern would be different from one individual to another. So that's a southern blot. Now, there are similar procedures that you can do using RNAs or proteins. So for example, if you wanted to identify RNA molecules of interest, you could take, here's an example. Say you want to know what RNAs, if a particular RNA is expressed in a certain tissue. You could gather all the RNAs, say, from a kidney in a human being, and chop them all up with restriction enzymes, and gel electrophorese them, I think that's a verb, and then probe it and see if you find the gene of interest, and if you, well, it would be the gene of interest being expressed on a messenger RNA, but you're screening and looking for a messenger RNA. Now, this is a little silly, you guys, but scientists, we're an interesting breed. I, of course, am perfectly normal, but that's beside the point. My point is this. They said, well, Southern invented this. It's a Southern blot. So if we do sort of the opposite, we're going to look for RNA instead of DNA. Wouldn't it be cute if we call that a Northern blot? I know, but for real, that's what they did. So a Northern blot is looking for RNA. You can use a single-stranded DNA probe or a single-stranded RNA probe. You can do a somewhat analogous thing with proteins. Of course, you can't use a nucleic acid probe for proteins. You can separate proteins via gel electrophoresis, although they have different charges, so you have to be a little bit more careful. Transfer them to a membrane, but the probe has to be an antibody, a labeled antibody in this case. It's not the same as complementarity among bases exactly, but antibodies bind very specifically to proteins. And you guessed it, this is called a Western blot because we had south, north, then we got west, whatever. I'm not aware of an Eastern blot, but I'm sure someone will come up with it soon if it doesn't already exist. All right, so that's an important thing to do, Southern blotting. I bet you might do it someday. Let's talk next about analysis by restriction mapping. So a restriction map simply is a depiction of the relative locations on a piece of DNA of various restriction enzyme cleavage sites. It's like a roadmap of a particular piece of DNA, most useful usually when that piece of DNA has not really been characterized that much yet. So it's pretty easy to do. Why would you want to do it? Well, we didn't talk about this, we should have before. You can verify that you cloned the piece of DNA that you wanted to. Hold that thought for a minute. Or you could just map a region in great detail. If you don't really know the sequence, this is a good first step to sort of mapping out a part of the genome. But here's simply the idea. You want to make a map and say, okay, wow, look, there's an Echo R1 site there. There's another Echo R1 site there, which I did it wrong. And then over here, there's a Hindi3 site. Over here, there's a Bagel1 site. So basically, this is giving you, in a sense, sequence information, but you're using these sort of as markers on your little roadmap here. Eventually, maybe you'll zero in on this area and get some more information about it. All right, but that's the idea of a restriction map. Now, to actually do it, let's just use an example where, remember before we were talking about gene cloning, and we said, okay, you chop out the human insulin gene, put it into a plasmid, and there you go. But in real life, you don't want to waste time and effort if this didn't work out correctly. So you want to do a little analysis and make sure that, in fact, the plasmid that you made contains the human insulin gene. So how would you do that? Well, you can do is take your plasmid, isolate it back, and then cut DNA with either various restriction enzymes or combinations of those enzymes in different tubes. In other words, separate them. And then separate them in a different way by size on a gel, and the pattern you perceive should match the predicted one. So in other words, let me try and draw a little picture of this. Say you had your original plasmid. We said there was an Echo R1 site in it, right? And let's say now that you put your human insulin gene in there, we didn't use it to cut, but let's just say we happen to know for a fact that there's a Bagel 1 site inside the insulin gene, right? Now, wherever this Echo R1 site still exists. So in other words, say you isolate your plasmid and you digest it in one tube with Echo R1, right? Well, actually, I'm going to add one over here. Let's put a Hindi3 site here. Okay, the reason I did that is because we're going to do a mixed digest, put Echo R1 and Hindi3 in there. Here you're going to get fragments of two sizes, right? So on a gel, you might see that. If you do the same digest of EcoR1 and HindD3, you should get one fragment that's the same size as the smaller one, right? But now that you've inserted a big old human insulin gene in there, this other fragment should be a lot larger. And that will tell you that you did insert something. And you can, if you put a ladder next to it, you'll be able to know the size. If you know the size of the human insulin gene, you can have good confidence that you succeeded. Now, if you digest with EcoR1 and Bagel1, for example, the original plasmid is only going to be linearized, meaning it won't change its size, but the new plasmid is going to be cut at two places. So this is the idea of how to do it. In real life, it's not that difficult. It can become a little tedious and some math is involved. It's fun. What if you want to make a restriction map of a piece of DNA and you don't even know the sequence? You basically follow a similar procedure. You could have a whole gel with, I'm not going to write it all out, but all different restriction enzymes, different combinations of them, and you'll see different size fragments depending on the situation. I'm just totally making them up, but it becomes like a detective story, and you can use all that information to deduce, okay, well, if I digested with this enzyme and I got this pieces, and I digested with this and I got these, and then I put them together, you can start deducing a map, sort of like the one we drew on the page before, where you're going to have all these different spots where the enzyme cuts. And sometimes that's very good information to have. You might rather have the whole sequence, but this is a good starting spot. Okay. Lastly, in this part, we're going to look at, it's hard to overestimate the importance of this tool, and that is literally getting the exact base sequence of a piece of DNA. So you probably know, we've referred to this, and you've probably heard it just in passing, that we can, in fact, determine the nucleotide sequence of any given piece of DNA. There are a few ways to do this, but the most modern and popular method was made by a guy named Fred Sanger, who passed away recently. It's more often now called the dideoxy sequencing method, and it's based on the idea of using so-called dideoxynucleotides. A deoxy, does not have a 3' OH group. And the 3' OH group, if you understood what we talked about during DNA replication, is necessary to extend a growing chain during replication. So if a dideoxynucleotide gets into a growing chain, it causes chain termination. Now, you may say, what does that have to do with the sequence? But stick with me for a minute. You guys know I can't draw, but I'm going to try and draw a little nucleotide over here. So here's a 5', here's a 3'. So the base is over here. Check this out. If this was an RNA nucleotide, the 2' carbon would have an OH on it, right? I think you know this. I think we've discussed it. But the whole reason deoxyribonucleic acid, DNA, is called DNA is because it's deoxyed. So that's removed. So DNA has at the 2' carbon just a hydrogen. But DNA does have an OH group at the 3' carbon, which is absolutely necessary for new nucleotides to hook on to a growing chain. If you take that one off, now you've dideoxyed it, right? The first one was taken off to make the DNA, which makes it deoxy. Now you took another one off, and you've got a dideoxy. And no incoming nucleotides can attach to that base. But it's fine up here. It can get incorporated into a chain. So let's check out the brilliance of this. How do you actually do DNA sequencing? You take the gene or whatever DNA it is that you want to be sequenced, and you're going to use it as a template to do in vitro DNA replication using DNA polymerase. So we've seen this a couple of times. You're going to simulate in the PCR, polymerase chain reaction, you're simulating DNA replication in a test tube as well for a different reason. So you're going to have 4 different tubes here, and in each of them you're going to have template, a primer, all 4 normal dNTPs, DNA polymerase, and a small percentage of either DDT, dideoxythymidine, DDC, dideoxycytidine, DDG, or DDA. Now, There's only a small amount of one of the dideoxys in each tube. So what's going to happen is a series of what we call nested fragments is going to be created, each of which terminates at, say, an A residue, if that's the tube that you're thinking about. So in the tube that has dideoxy ATP in it, you will have a nested bunch of fragments. Each of them ends with A. Then you still keep those 4 tubes separate, run them out on a high resolution electrophoretic gel, and you can literally just read the sequence 5 to 3' from the bottom of the gel. Let's look at a picture because I know if you didn't get that yet, you will now. So here is the area of interest. I mean, usually it would be a lot bigger, but we just want to use this for our information. We're going to sequence it. So first we've got to denature it, and we're going to only focus on one of the sides now. And only one of the sides will be used because we're going to make a primer for that particular side. So we want to get this sequence right here. We need to put a primer in, and the reason is, as you know, DNA polymerase cannot start a chain, it can only extend one. Now you might say, see, the primer I made on the picture is GGA because I knew the beginning was CCT, but you might be saying, well, what if you really don't know? Well, sometimes you have partial sequence information or you want to extend from a place that you know. In that case, you could make a targeted primer like this. If you know nothing, sometimes you can just make a bunch of random primers so you know that it'll stick somewhere and then extend, and that's often just as good. But I just wanted to show you, look, we wouldn't have known any of this sequence, but I'm showing it to you now because I want you to understand how we're going to get the results. We got DNA polymerase, we've got all 4 of the NTPs, but very specifically in one tube, we've got one of the 4 dideoxy NTPs, probably in a ratio of about 1 dideoxy to 99, 1 out of 100 of the regular nucleotides. So after annealing the primer is going to extend, right? But here's the really exciting part. So we're going to go extending the primer. Now, in the tube that has a little bit of GDP in it, right? 99 times out of 100, when the primer gets extended, right, a normal G will get put in there and it'll just keep going. But 1 out of 100 times, and this is going on a lot, a dideoxy will get put in there and that will cause chain termination. So everywhere there's a G, you're going to get a nested, you're going to end up with a nested series of fragments, each of which ends with a G. In the A tube, you're going to get a similar thing, but each of them ends with an A, or with a C, or with a T. And when you run those out in 4 separate lanes on the gel, it's simple, guys. You can read it. Now, because look, the shortest fragments are going to be, remember, this gets extended in the 5' to 3' direction. So the shortest fragments, which are going to travel the furthest down, because they're small, in a gel, are going to be at the 5' end. And you can literally just read up the sequence. Look, check it out. The first fragment we see is in the C lane, so it's a C. Then we see A. Then we see T. I mean, once you get it to this point, you literally can just read off the sequence. Sometimes it could be 1,000 long. You could just read it off. There are machines that do this for you as well. They scan the gel and they look at the density and it just reads you off the DNA sequence. These are high-speed sequencers. But it's very important to understand why you need the dideoxys in there and why also the ratio is important. If you had think of this, if you put all dideoxy G in that tube, well, always it would end with that first fragment, and all you'd be able to sequence is the first G that's there. We want to have a whole load of fragments so that we can read them down on the gel and find every place a G is. So that's dideoxy DNA sequencing. I hope you enjoyed that because I certainly did. And if you are going along in order, the next session focuses on, we've already sort of implied it, but the applications of all of these techniques to exciting and modern things. I'll see you then.